Welcome back, folks. This is going to be a video lecture on chromosome variation. A pretty straightforward analysis of chromosome structure and variation. Let's get started with the agenda. A quick review of what a karyotype is. Then we'll move on swiftly to talk about deletions and duplications, and their cousins, inversions and translocations. And then finally, we'll end today with an understanding of ploidy, both Euploidy and Aneuploidy. And then just to finish off, we'll have a look at molecular mechanisms that can result in variation in these chromosomes within a cell. So let's get started quickly. The insert that you see over here is a picture of somebody that we should give credit to, but we don't. This is Jemser. Gustav Jemser. He was instrumental in developing stains for chromosomes. You can see the chromosomes below have been stained, and they have banding patterns that you can just about see in this particular figure. And if I point them out, you would see that there are different bands along the length of chromosome number 2, for instance. And those banding patterns are good in identifying chromosomes. So let's have a look at what a karyotype is. So a karyotype is basically that. It's a picture, a micrograph of chromosomes taken at a point when they're visible inside the cell. If you recall with the cell cycle, only during parts of the M phase are chromosomes visible. So late prophase is when they start condensing, and then they become really pretty visible between metaphase and anaphase and the beginning of telophase. So these particular chromosomes have already been replicated at that point, and up until anaphase, they'll stay replicated in mitosis and meiosis 1. And that's what you're looking at here. So chromosomes are classified according to 3 things. Their size. So you can see the largest chromosome in the human case is chromosome 1, and then followed by chromosome 2, 3. So they're placed in order from large to small. And the smallest chromosome in the human situation would be chromosome number 22. And then, of course, the sex chromosomes are placed last in the karyotype. Now, in this particular figure, you can see there's something different about the number of X chromosomes. So this particular karyotype was taken from a mutant cell that had an excess of X chromosomes. We'll come back to that in a different video. So once you organize the chromosomes by size, the next thing is to organize them according to their shape. So chromosomes can be classified according to the position of the centromere. The centromere, of course, is the central body somewhere along the chromosome that holds the sister chromatids together. And it's also visible during non-replicated chromosomes as a heterogeneous spot, a heterochromatin spot somewhere along the chromosome that serves as a structural function to move the chromosome during cell division. So chromosomes such as chromosome number 1, you can see, will be called metacentric. So meta stands for middle. So the centromere should be in the middle of the chromosome. The term submetacentric would apply to chromosome number 2. So the constriction is almost in the middle, so around the middle, okay? And then we get acrocentric. Acrocentric would then be something that's towards one end, definitely, than the other. So here we have some acrocentric, and here we have some acrocentric, and there we have some acrocentric chromosome. And then finally, we have telocentric. And telocentric normally means near the end of the chromosome. So we have 3 pairs of human chromosomes that are telocentric or telocentric. So these are important classifications that you find for chromosomes. One last thing, when you have something divided into 2, a top and a bottom, and one side is slightly bigger, but it doesn't matter by how much, but one side is slightly bigger than the other. The bigger side goes on the bottom and the smaller side goes on top. So if the top of this chromosome that I've drawn here was smaller than the bottom by doesn't matter how much, then we will call that the P arm, for French for petite. And then the next letter in the alphabet will be Q, so PQ. Okay, so here we have the P arm of chromosome 8, and here we have the Q arm of chromosome 8. Now, sometimes chromosomes are both the same size and they're the same shape. So how do we classify those chromosomes? Well, we go to the third level. And the third level is where Gemza comes in. So he developed a staining pattern, and that is still used today to stain the chromosomes so that they produce bands. And the banding pattern can change depending on how compact the chromosome is. So highly compact chromosomes have a slightly different banding pattern than more loose chromosomes, right? So Gemza staining can be used to then identify different chromosomes, because each chromosome in the human should have a similar staining pattern. So let's continue our journey, and we're going to look at some mutations of chromosomes. So mutations are simply changes from the normal wild type condition. And this can apply not just to individual locations within the gene, nucleotides, but also at the level of the chromosomes. So the term mutation is quite broad, and it means any departure from wild type. So what type of mutations do we encounter with chromosomes? There's 4 major types, 2 of which are listed on this slide. We have duplications and deletions. So let's start with deletions first. Just show you quickly, this chromosome pair here, they're not really a pair, it's the same chromosome seen before the mutation and then seen after the mutation. And the area that you see in purple, the wedge, has disappeared. So this is what you find in the wild type, and in the mutant chromosome, that region is missing. Now, naturally, chromosomes contain lots of genes, and any region that has a significant size and that's missing can result in the deletion of all the genes therein. So in this particular case, it would seem like that this mutation has resulted in a lot of genes being removed from the chromosome. And the chances of survival of the individual cells that contain these, or the fertilized egg that contains these, either way, will be dire. And the chances are that that particular cell or the organism won't survive. Okay, so loss of genes is considered lethal in most cases. So deletions can encompass small regions or large regions. So some of them may be visible like this on the karyotype, but others may be too tiny to be seen with microscopes. Therefore, you may need an electron microscope or some type of genetic analysis to determine those. But regardless, a deletion is some missing information from a chromosome. Deletions can be terminal, where they can happen in this region here, or they can be interstitial, as in this case here. So the end of the chromosome could be lost, or you could have an interstitial. Now, the difference between the two is that terminal deletions, these types, they just require the DNA molecule, the DNA double helix, to break once across both strands. So here's your DNA. And essentially, a chromosome is a single huge DNA molecule wrapped around proteins, histones, and compacted to this shape. So what happens in terminal deletions is you get a break across the DNA helix, across both strands, and the smaller piece, the one that doesn't contain the centromere, can be lost. Whereas interstitial deletions, they require 2 breaks in order to lose that piece of DNA. So in the second example down the bottom, we would have 2 breaks, and then that piece will be lost. So 2 cuts in the DNA. just to finish off this quick section, one of the most commonly cited types of genetic mutation when it comes to deletions in most resources is a syndrome called cri de chat. Cri de chat, it's a French term. It means cry of the cat. So when these infants are first born, you can identify this particular syndrome by the sound coming from the newborn. It sounds more like a cat than a human baby. And that, unfortunately, is an indication that this individual has a deletion. And it's normally on chromosome 5, human chromosome 5, and it's going to be the P segment of chromosome 5. We go back to this figure here, it'll be this region of the chromosomes. That will be one of the two homologous chromosomes that will carry the deletion. So that's cri de chat. So let's move on to duplications. And as the word suggests, under normal circumstances, as we'll see down here, the wild type chromosome has a region that's been duplicated. And again, these can be small or large, and depending on the size, they can have consequences. And we'll talk about consequences on the next slide. So the region is repeated, and everything within that region, all the genes within that region, and all the other control elements within that region of the chromosome would also be repeated. Okay, and the result is a duplicated chromosome. So what causes chromosomes to become duplicated? So let's quickly make some space over here, and we'll try to relay that as a graphic. Let's go back to let's choose a different kind. Let's go for the orange pen, just for the sake of changing things. So what causes duplications? Well, normally what happens is repetitive sequences, which we have scattered all over our genome. There's a mistake in the alignment of homologous chromosomes or sister chromatids. And then when there's a crossover event, that can cause a reshuffling of the regions of the chromosome. So imagine that we have a chromosome like this, and it's supposed to line up with this homologous chromosome like this, so that we have perfect alignment, and then you can get crossing over between the DNA. What happens in duplications, and the most common event, is that the two regions, they misalign. So one chromosome would be like this, and the second chromosome would be like this. And then there's crossover. And the reason for that is there's a region probably here that matches this region here and this region here, right? And what happens is that the two regions misalign such, and then there's a crossover. And then the best way to follow this is to draw a line. Let's choose a different color, and then draw a line and cross over and follow the other way. And now you'll see that this segment and this segment appear twice on the chromosome that I was drawing in blue, and then the complementary chromosome would be like that. And that chromosome there would probably, in this case, have a deletion. So normally deletions and duplications go hand in hand if it occurs by this particular crossing over misalignment, okay? So that's what we get as a result of having these repetitive sequences. Now, most repetitive sequences are not encoded in the human genome directly, but they could be placed there by transposable elements and retroviruses and things like that. So parasitic DNA would come under that title too. So in many textbooks, the term that they can use to represent this type of misalignment will be a non-allelic homologous recombination. And that's exactly what you see up here, okay? And now you know how you can get gene duplication. Now, gene duplication may not be a bad thing. If certain genes are duplicated on a particular chromosome, then that chromosome, as it travels through the species, will have copies of genes. So you'll have the primary gene and the copy. the good thing is this becomes great fodder for evolution. So evolution can start working on the copy without worrying about changing the biochemistry of the primary gene. So you have two copies. The original one keeps doing its original job, but the copy can now vary and tinker with itself to produce new functions or different functions. And that can lead to gene families. So gene families are simply genes throughout the genome that are related to each other through sequence and function. And the reason they're related to each other is because they came from duplicated genes by a mechanism such as this. And the classic example that we use again in genetics textbooks is globin, the globin gene family, right? Myoglobin would have been the original protein that allowed our ancestors, distant ancestors, maybe primitive animals, to carry oxygen in their bodies. And then from that, gene duplications seem to have happened twice, two major times. And the first time led to the formation of the beta globin type genes. And then from that, we now have the alpha and the beta globin genes, right? And humans have 14 of those genes, and they are active in different tissues and also at different times during our life cycle. But that's a different lesson. But the globin gene family has to be giving credit to gene duplications for its present day existence. Okay, so when you have genes that perform the same function and they come from the ancestor, we call those homologous genes because they arose in the ancestor before speciation, and now this species has it, and potentially the sister species has it too. For instance, if this type of duplication took place in pre-humans before Neanderthals and modern humans, and then Neanderthals would have inherited this type of duplicated condition as well as human beings. If a species has genes that are copied and that they are not present in other species around them, their ancestors or sister species, then we call those genes paralogs. Okay? And then we go on to finally look at copy number variation. So copy number variation simply means chromosomes that have the legacy of a past duplication, and that duplication has resulted in a gene copied next to itself. So you have two genes the same sitting side by side on the chromosome, and that's called a copy number variation. Now the copy number can vary from 0, which is the gene is missing on that particular chromosomes. That could be detrimental, but it depends on the nature of the gene, or the normal condition, which is 1 copy of the gene per chromosome. And then beyond that, you can have 2 copies, 3, 4, 5, 6, 7. In fact, believe it or not, a significant proportion, up to 10% of the human genome, contains copy number variation in genes, right? And that is known as a segmental duplication. So in this case here, if there's a gene right there, this is just exaggerating, if there was a gene right there and that gene was duplicated, then this region there would be called a segmental duplication. A segment of the chromosome has been duplicated. So hopefully all these terms make sense to you. Let's move on and rapidly look at their cousins, inversions and translocations. So let's start with inversions. So believe it or not, the bottom figure here, this is demonstrating an inversion. And you can see the original order of the chromosomes would be, say, region 1, 2, 3, 4, 5, 6, 7. And you can see there's been a cut here and there's been a cut there. And the chromosome has been flipped and then reattached to the 2 arms, right? So in this case, we'll get 1, we'll get 4, 3, 2, 5, 6, and 7. So this region has been inverted, right? So therefore the word inversion. So that's an example of an inversion. Now, if the inversion occurs at the end of the chromosome, for instance, this part was inverted, then we call that a pericentric inversion because it doesn't involve the centromere. It does not involve the centromere. In a paracentric inversion, that would include this region here. And that would have to include the centromere. So para means around, centric means centromere in this case, and peri means away from the centric region, which is the centromere. So the example that's given here in pink would be a pericentric inversion, pericentric, right? Because it doesn't involve the centromere. So that's a simple explanation of that. Now, we may remember from a different chapter that when you have genes missing, that can be very detrimental to the organism. And you can see here, there are places where genes can be missing in duplications and deletions. So deletions result in genes missing. Duplications result in extra copies of the genes. You may think that there may not be much of a consequence, but when you have missing genes of the same degree as you have duplicated genes, say you have 10 genes duplicated, or in a different individual, 10 genes deleted, of the two, the deletion is more dangerous, is more detrimental than the duplication. Although both are bad, right? Because as we'll learn later on, they upset the balance of biochemistry amongst all the rest of the chromosomes and this region that's duplicated or deleted. So any kind of duplication or deletion, especially if it's significant, that involves a significant part of the chromosome, can be lethal in many types of creatures, right? So animals don't tend to tolerate these conditions as well as amphibians, for instance. Amphibians seem to have a biochemistry that can tolerate larger deletions and duplications than mammals. If you move genes by cutting the DNA and placing it in a different region, then maybe that region will dictate the compactation of the chromosome. So if you take a region of a chromosome that's shut down, and then you place it somewhere else where that region is active, then the chances are that new piece of chromosome that's moved from the original place to the new place will also be active, and the genes will be turned on when they shouldn't be. Therefore, we call that a positional effect. And that complicates our analysis of gene migrations due to these types of mutations. So, you know, it's a very complicated understanding of how chromosomes work, but it is a fact. A heterochromatin versus euchromatin. Are you taking some heterochromatin and putting it into euchromatin by these type of mutations? If that's the case, then the consequences for the cell will be biochemically upsetting. So we call those positional effects. Depending on the position, it can have a different effect. So let's move on to talk about something that can happen during inversions. So when you invert something, for instance, let's take the example of the blue chromosomes down here. Let's tidy them up a bit so we don't get confused with all the different numbers. Let's get the eraser going, and then I'll just erase my little labels that I put on here. Okay, so we're now ready to look at this phenomenon, right? Inversion heterozygote. Now imagine that these two, imagine there were two chromosomes now like this, the original one, the wild type, and then the mutant. And they were to pair up with each other during meiosis 1, right? When homologous chromosomes are paired up into a bivalent. Now, let's redraw these chromosomes in that situation. I'm going to turn them to 90 degrees so we can see them better. So let's take the wild type chromosome and draw it like this. Okay, there you go. And now, just remember, it's in meiosis 1, so it would have been replicated in S phase. So we need to have sister chromatids. So let's do this. So here's the same centromere. And then from here, we have a sister chromatid. And from there, we have a sister chromatid. So this looks like an X. Now let's take the mutant chromosome and line it up with it. Okay, so the mutant chromosome will also be duplicated at this point. and it will have its centromere here, and then it would have its two arms. Okay, one arm for one sister chromatid and the other. Now just remember this, that we're looking at this region here now, and if we number this 1, 4, 3, 2, 5, and 6, remember we had that inversion between 4 and between 1 and 2 and 4 and 5, right? So this the long arm of the chromosome, the Q arm, has a inversion, whereas the wild type chromosome still has the original configuration of segments, and so does the sister chromatid, right? So the two chromosomes on top have the same orientation, and the two chromosomes on the bottom have the mutant chromosome. So we're going to color these two in green because they're the wild type, and then we'll use red or some other color to indicate the mutants. Let's use a felt tip pen because it comes out better. So here we are, right? So those two sister chromatids those two arms of the sister chromatids are mutant. These pair of homologous chromosomes have no problem now lining up. So they line up here all along their lengths. Everything is the same. And then they line up in segment 1. Now segment 2 and segment 4 are in different alignment, right? So segment 2 here wants to line up with segment 2 there. And segment 4 there wants to line up with segment 4 here. And guess what? The chromosomes are flexible enough to permit that. So what shape would you end up with? You'll end up with a weird shape where the chromosomes are doing some kind of contortion. And that's exactly what we get. And we end up with that term there. We end up with a term that will lead to the formation of an inversion heterozygote, right? So let's look at that. So let's redraw these chromosomes in that new configuration. Okay, so let's try to put them together. Let's have the bottom chromosome form a loop in order to accomplish that task. So let me get my pen back out again of the right color. Let's use red for the skeleton. So we can draw one like this, one pair, right? There you go. And then the centromere will be here. Okay, and we can divide this into 2 to represent both of the sister chromatids. So basically, I'm moving this down here. So let's use the felt tip pen to color that, the appropriate color. That was green. All right, there we go. Okay, and the green can continue down here. And now let's marry that to this bottom homologous chromosome. So the bottom homologous chromosome will look like that. And let's color that in so we can see that in red. So there it is. Oops, red felt tip pen, sorry. Here we go. There it goes like that. Then it comes down and it goes like that. And if you look carefully, let's use black to label these down. So segment 1 will still be here. Segment 2 will be there. Segment 3 will be there. Segment 4, segment 5, and segment 6 of the top green homologue. And now the bottom homologue would be like this. And then this part here will be region number 4 will be here, 3 will be there, 2 will be there, and 1 will be there. So as you can see down here at the bottom, in this manner here, you can get homologous chromosomes forming a bivalent by doing this acrobatics. The next question is, what would happen once you get crossing over? So if you get crossing over, and I'll use a blue color. So this is very important for your assessments. If you get crossing over in this region right there, what would be the outcome for this type of meiosis? What would be the outcome for the 4 types of haploid cells that will be generated once the entire meiosis is complete? And that's what you need to figure out. So I'll help you here quickly, and we'll use the space above because I need the extra space. So let's get rid of some stuff that we don't need. And let's get rid of this for now. And let's get rid of this arrow. And let's get rid of anything that confuses us. Okay, now that we've drawn our chromosomes, what we need to do now is to figure out what happens subsequent to meiosis. We need to identify what the 4 chromosomes will look like after this crossing over. So what I've done, I've tidied up the slide so that we can draw above what the outcome is. So let's do that. So we're going to assume, let's use my pencil first. We're going to assume that we have the top chromosome here of the green homologous pair. Then we have to draw the bottom chromosome of that homologous pair. And then we have to draw the 2 chromosomes that were involved in the crossover in this particular event. So we have 4 chromosomes to draw. Let's take the easy ones, the ones that were not involved with the crossover. So those are the easy ones to deal with. So at the top, we will have a chromosome with its centromere right here, right? The original one. So this will be this wild type chromosome, and it'll have its centromere there. And from the centromere, we can number it as we had in the original case. 1, 2, 3, 4, 5, 6. So that's a wild type chromosome with no inversion. Now let's look at the bottommost chromosome, this one down here. That will be an unaffected mutant chromosome, an inverted chromosome, right? So let's draw that. So that one down there would be like this. And it too would have, it too would have its centromere right there. But it would be the same as the mutant type, okay? So it would have region 1, 4, 3, 2, 5, and 6. The reason for those 2 chromosomes having their original configuration is that they were not involved in the crossover. And that's the example that we're giving, okay? But in your case, in a separate question, they could ask you what would happen if the top and the bottom sister chromatids of homologous pairs are involved in a crossover. But in this case, we're assuming the closest 2 sister chromatids are involved in crossover of the homologous chromosomes. All right, so here we go. So what outcome do we have with respect to the 2 sister chromatids that were involved in a crossover? So let's put that down. So now the crossover took place between region 3 and 4, right? So let's draw this in a way that we can relate to it. So we have this chromosome here, and then we'll have, I'm just going to save time, and then we're going to have the second chromosome here. And then for the other one, because of the crossover, a part of that would carry something else, and this part will carry something else. Okay. So is that the right thing to do? We'll find out in a second. So let's just follow one of the chromosomes from right to left, and then we'll label this one up accordingly. Right? So let's start with this chromosome here. So we start over here. So we're going to start right there, and there's the centromere. Now that corresponds to this spot here, right? So that's in the same place. Then we have region 1. So region 1 is in the right place. So that will be right there. Then region 2, as we come around here, is in the right place. So region 2. Region 3 is in the right place. So region 3. But now see what happens. Now we shuffle down to the red, and then we go around here. So we have 4 as we come down here. And then we have 1 again. And then we have another centromere. And then we have the rest of the red. So in this case, we would find this. A chromosome, because of crossing over, that's generated later on, would have 2 centromeres. So that is called a dicentric chromosome. So the final piece of the puzzle that we have left is to figure out what piece of DNA is left over once all the parts have been qualified. So we find that we have the following, and I'll explain that in a second. So what we'll find is that we have a region of green with a region of red, like that. And there's no centromere. So what does that correspond to? Let's use our pencil to dictate that. So we're starting on the green here, and we're following the green as we go around, and then we cross over, and then we come down this way, right? So let me use a different color pen to help us with that. Let's use a blue color. So we're going along here, we go up here, and then we come down here and round there because of the crossover. And that leads to our friend here. That will be your acentric chromosome. And an acentric chromosome will be lost when the nuclear membrane reforms at subsequent stages of cell division. So a lost chromosome is then degraded by enzymes in the cytosol at that time. The chromosome with 2 centromeres will have a hard time being resolved, and it will most probably sustain a random break between the 2 centromeres at some point. Therefore, it could break there, there, or there, or there, and that will then give those particular segments to the daughter cells. And it's unlikely that they'll be viable. Okay, so you can get 2 gametes, this one and this one, that should still be viable, even though the bottom one has an inversion. Depending on the positional effects, that could be viable. The top one is wild type, so that sperm would be viable, or egg. But the middle 2 definitely would not be viable unless both centromeres of this chromosome ended up going to the same pole during cell division. So pretty complicated situation. But guess what? Treat it like a Sherlock Holmes mystery, and you'll be surprised how easily you can figure this out. One last thing. In my particular example here, we were not involving this region. We were not involving the centromere. So these were pericentric inversions. What you need to practice, what would happen if this region was in here? What would be the outcome? And once you can do that, then you have mastered this concept of real life mysteries. So try that, please. I've taken the liberty to make some room for drawing again on this slide. And now we're ready to move on to translocations. So translocation simply means that a segment of chromosome has broken and has attached to a different region of the genome. In the figure that you see here at the top of the slide, we're dealing with chromosome 13 and chromosome 10. So chromosome 13, in this case, is going to be this one here, even though it looks larger than 10. That sort of violates what we learned about before. So that's not true. So whoever drew this figure that I borrowed, it looks like it's a figure from Spain or somewhere like that. Regardless, doesn't matter. The principle is the same. But you see how you can identify errors in figures if you look carefully. Regardless, let's go on. So this region is normally present in chromosome 13 in this case, right? And this region here is normally present in chromosome 10. What we find after the mutation is that this region from 10 has now moved into 13, and this region from 13 is now present inside 10. So that's called a translocation. So let's look at the different categories of translocation. So simple translocation is where one chromosome loses a segment or set of segments, and they end up being attached to another chromosome. So simple one-way migration. Pretty difficult to happen because of the telomere, but it can happen in certain rare circumstances. So let's draw that. So we have a human chromosome, say chromosome number 1, and then we have a human chromosome, say chromosome number 12. Suppose this part of chromosome number 1 and breaks off and joins the tip of chromosome number 12. Now we will have a shortened chromosome 1 that's missing its tip, and then we'll have a longer chromosome 12 that now has an extra piece added to it. So that piece there has moved down there. So that's called a simple translocation. The opposite of that is what we see up here in this figure, and that's called a reciprocal translocation. So one chromosome has a break, another chromosome has a break because of the same event, and those fragments, those segments are then switched over and then pasted back onto the chromosome using DNA repair mechanisms. So that's called a reciprocal translocation. The most common are the reciprocal, because simple requires a bit of biochemical maneuvering to deal with the telomere on the end of that chromosome. So that's what you need to understand right there. So what is a balanced translocation? A balanced translocation. So we're going to look at that now. So a balanced translocation is simply where the total amount of genetic material is maintained between all these maneuvers. So the size of the genome doesn't increase or decrease. It's just a movement from one place to another. In an unbalanced translocation, DNA is either duplicated during these breaks and maneuvers, or more commonly, DNA is lost, deleted. And that can cause those segments that are deleted or duplicated to have an additional effect other than positional effects. So once again, because you're moving this segment to a new chromosome, maybe this segment of chromosome 1 was highly active in the wild type chromosome. And maybe this region of chromosome 12 is semi-active. By putting this region here, you're now introducing it into a region of semi-activity. Therefore, the whole region of chromosome 1 that's now moved onto the top of chromosome 12 may be diminished in its general biochemical activity, therefore reducing the gene product and changing the biochemistry of the host cell. So we have to keep an eye on that. In addition to that, in an unbalanced chromosome, pieces of DNA may be lost either from the target chromosome or the donor chromosome, the recipient chromosome or the donating chromosome. And that loss of information now becomes critical. How much information did you lose? Which genes are involved? How critical are those regions? And that will then affect the outcome for the individual concerned. The classic example that we give is Robertsonian translocation, and that involves human chromosome number 14 and 21, swapping parts. So what happens is, I believe the long arm of 21 is moved and attached to the short arm of 14, making a 14 centromere 21 hybrid. And then at the same time, the short arm of chromosome 14 is combined with the small arm and centromere of chromosome 21. And that's the Robertsonian translocation, first identified by Dr. Robertsonian. So he gets credit for that. So once again, the question is, once these types of maneuvers have taken place, what happens in meiosis when the chromosomes come together with their wild type compatriots? And that is going to be resolved depending on what parts of the chromosomes are involved. So we get something known as a translocation cross. So the two pairs of homologous chromosomes, they come together and they have to fit together in a form that the arms with their different segments line up. And that looks like a cross. Let me draw that very quickly for us. So let's get rid of this. And now we can draw that here. Okay. So let's use felt tip pens again. So imagine that we have a pair of chromosomes. So these are the sister chromatids. And on one of them, there's a translocation. So this one will bend this way because it's the original. And then let's draw the other one with a translocation. Let's use green. So this one has a translocation. It goes up. And then that pairs with the wild type chromosome there. And then we have the other chromosome with the translocation pairing up in such a fashion. Okay. And the centromere in this case for this chromosome will be over here. And for the other chromosome, maybe there. So we get this translocation cross where the different segments with the different numbers line up. So that's another challenge for you folks. Just look on the internet or your textbooks and you'll see how this comes together. And then the question is, how do you resolve this? How do you pull it apart if you do get a crossover, say there, or a crossover here, or maybe a crossover there, or maybe a crossover there. So those are the challenges. So many possibilities depending on the position of the crossover. Let's turn our attention now to look at ploidy. Ploidy is a two-part term. If it involves individual sets of chromosomes, whole sets, then we call that euploidy. If the mutation involves individual chromosomes within a set, then we call that aneuploidy. Aneuploidy will be discussed on the next slide. Here we're going to go to the former example and talk about whole sets of chromosomes, right? So the mutation is ploidy, the type of mutation is aneuploidy, and in this case, we're looking at sets of chromosomes, okay? So let's look at the human case. So with higher animals, mammals for instance, we find that we're mostly diploid. So we have 2N. That means for humans, we have 46 chromosomes in somatic cells. Therefore, N would be 23. And we find N in gametes, okay, sperms and egg. For amphibians, we find that they can have large deviations from this particular schema. Why? We're not sure. Amphibians are animals, but they're not mammals. So there could be some biochemical disparity between the branches of the tree of life. And we also find that plants with their simpler tissue organization, they can also tolerate changes in chromosome sets. So let's keep that there then, right? So we have plants and animals. Those are the most common that experience euploidy. We can find in the wild plants that have 4N equals something. So they have 4 sets of chromosomes. That means they have 4 chromosome number 1s, 4 chromosome number 2s, 4 chromosome number 3s, etc. What's the benefit of having that? Well, we find that on average, creatures that have more sets of chromosomes, they tend to be more sturdy for some reason. They tend to be bigger. They have bigger organs like flowers and fruit, or they have bigger size. So you can compare, in some cases, 2 frogs side by side. You can compare the diploid frog with the tetraploid frog, and you can see the tetraploid frog is a tongue twister. Tetraploid frog is twice as large in some cases as its cousin, which is diploid. So we can use these words here: diploid, haploid, triploid, tetraploid, etc. So ploidy is the term that we use. When it comes to sterility, we can make use of the fact that in certain manipulations, we can generate tissue that is plant tissue mainly, that is triploid or pentaploid. And that leads to fruit that is seedless. So seedless bananas, seedless watermelons, seedless grapes, etc., etc. So how does this work? Well, if a plant breeder was to take one plant cell that had, for instance, 2 large chromosomes in its male pollen and breed that with another related species of plant female, the egg, that had one chromosome of that same type, then the offspring would be triploid with respect to that chromosome, triploid. Now you can see what happens. When this particular plant reproduces itself, it has to undergo meiosis to produce both pollen and eggs. And that's where meiosis has a hard time. What normally happens, if we relay this to you in a different fashion, what normally happens in this case is that the system gets stuck with respect to meiosis. So when we have the spindle formed inside this new progeny, and it's about to produce the F2, we have one pair of chromosomes line up, because they're duplicated now. One pair of homologous chromosomes line up like this, but we have an odd chromosome with no partner. And in this case, meiosis will do its best to segregate those chromosomes, but we always end up with cells that contain an aneuploid. Aneuploid. The aneuploidy means an odd number of chromosomes, and those particular gametes are not viable. Therefore, the parent plant in which this is taking place is unable to form seeds. So the situation that you're looking at here is just involving one chromosome. Imagine all the chromosomes inside the cell were doing exactly the same thing. Then it's unlikely that you'll ever, because of permutations, get two gametes that are viable. Therefore, there will be no seeds produced. The only way to market these particular types of plants is to take cuttings and grow the cuttings season after season. And that way, you're guaranteed to propagate the plant. As you may recall from another video lecture, we were talking about haplodiploid species. At that time, we mentioned the bee, the honeybee. There are many other species of insect that are haplodiploid. That means the male of the species is haploid, and the female of the species is diploid. So females are produced through sexual reproduction, and males are produced through unfertilized eggs, i.e. cloned. So ants and certain types of wasps are also party to this particular genetic process. So it does happen. So there are animals that are other than diploid, and they do exist. So remember that. And finally, let's go to an odd case of polyploidy that can happen in certain tissues of the body of an animal or a plant. So Endo Polyploidy is where, through mitosis, certain cells capture all the chromosomes, or there's a special process at work, increasing or decreasing the number of chromosome sets in that tissue. So endo means inside the tissue of the organism in random places. And polyploidy means that the number of sets of chromosomes is abnormal. So the great example that all genetics textbooks use is that of Polythene Chromosomes. The saliva glands of a Drosophila fruit fly are very important in producing lots of saliva. And the cells that make up the salivary glands of that organism undergo a very unusual method of generating more gene product from their chromosomes. And the way they do that is to undergo cell division, M-phase mitosis, without cytokinesis. So what happens in there is that a chromosome is copied through S phase, and then it's copied again through S phase, and again, until you get over 1,000 DNA strands all attached to the same structure. And that structure is called a polythene chromosome. Polythene. What does it do? Well, it allows huge amounts of gene product to be generated for the benefit of those particular tissues. Now, when this process happens along all 4 chromosomes in the fruit fly, so fruit flies have an N number of 4, you get some complex structures forming within these tissues. And those are called the Chroma Centers. So of the 4 chromosomes, they all come together like this to form a structure where the centromeres of the chromosomes are all compacted together inside the cell nucleus, such that they look like an octopus. But instead of having 8 legs, this particular entity has 5. So that's what a Chroma Center is. The middle there, where the overlap takes place, is actually the Chroma Center. And it's a fascinating thing to see inside the salivary glands of these flies. Let's move our attention on to a cousin of Ploidy, Euploidy, called Aneuploidy. And the major difference is, as in the previous example we just finished, where entire sets of chromosomes vary, in this particular genetic mutation, single chromosomes or a few chromosomes within a set are either present or missing in excess. Now, you may not think that this would cause a problem. Having a small extra chromosome amongst all the others shouldn't make much of a difference. But nature is so wonderful, it's balanced all gene products with each other. So there's a harmony going on inside a cell through natural evolution. Everything is balanced. Remember, even the tiniest chromosome inside an organism can contain hundreds, if not thousands, of genes. And the products of those genes may then be curtailed if there's a missing chromosome, or expanded if there's extra chromosomes present. To best illustrate this, let's look at an example that we find in humans that most of us are familiar with. That would be Down syndrome. In Down syndrome, the individual has one of the smaller chromosomes present in 3 copies. So that would be 46 chromosomes plus 1. So that would be 2n plus 1. And that's what we see there. So the 1 indicates that there's an extra chromosome amongst the diploid set of chromosomes. So we have 3 copies of chromosome number 21 in that individual. And what does that do? That leads to mental and physical changes in the individual that can also have an impact on fertility and life expectancy in many cases. The situation is so complex and so dire that anytime you have extra copies of chromosomes, the offspring is challenged. And there's a reduction, as I said earlier, in the viability of that individual. So in most cases, those individuals are not born. They're miscarried early on during pregnancy, and they're not delivered. Somebody's estimated that 50% of miscarriages take place because of chromosomal number abnormalities, mainly due to aneuploidy. So let's look at how this system pans out. So you could have an egg of a mother and the sperm of a father, and one of these two will carry an abnormal number of chromosome 21s. So maybe the sperm is carrying 1 copy and the egg is carrying 2 copies of chromosome 21, plus all the other chromosomes in their haploid concentrations. So when that fertilization takes place, then the individual is born with 3 copies of chromosome 21, and we call that trisomy 21. So in humans, we have seen individuals born only under 3 conditions when it comes to autosomes. We have chromosome 21, which can be tolerated in extra amounts. We have chromosome number 18, which can also be tolerated in excess amounts. And then we have chromosome number 13. If you notice, these are very small chromosomes. So the higher the number, the smaller the chromosome. And they carry a limited number of genes compared to the large chromosomes. And the large chromosomes cannot be tolerated at all in excess amounts. So these 3 conditions are present in birth of humans. Unfortunately, the bottom 2, they do present with fertility problems that can reduce the life of the individual, and indeed they do. Down syndrome children, if taken care of properly, could survive a normal life expectancy. So there's the power of imbalance in the number of chromosomes within a set. But we haven't finished yet. We have to look at the names of this. So this is called Down syndrome. Chromosome 18, when present and identified inside a human being, is called Edward syndrome, Edward. And the last one is called Patel syndrome, right? So those are the syndromes that you should be familiar with. Just to finish off, as you learned in a previous video lecture, the sex chromosomes can be present in abnormal amounts, and they are tolerated more because of the way that they are matched up with the autosomes. So their biochemical behavior is slightly different to the autosomes. So we saw examples of XXX and Turner syndrome and Klinefelter syndrome in males. So remember those, please. Those are also examples of this. Let's take a trip back slightly and look at what the cause of this is in one parent or the other. Suppose the sperm of the dad is going to deliver 2 copies of these chromosomes. So what happens is that during the formation of the sperm, as the sperm undergo meiosis from the haploid tissue, in one case, both chromosomes are pulled towards the same pole of the cell, and none are pulled to the other pole. So this process, as we'll discuss next, is called nondisjunction. Nondisjunction. And the sperm that gets 2 is viable, and the sperm that gets none is not viable. So this will affect the sterility of that individual. And there's many ways that sterility, human sterility, is affected. But in this case, you can see that half the sperm in this particular example from this nondisjunction would be non-viable. But nondisjunction is very, very rare inside humans. So it's not likely to affect all the sperm. In fact, just a fraction of the sperm will be affected in this fashion. But that doesn't matter. If this particular sperm fertilizes a normal egg with one chromosome 21, then you will get a trisomy 21. And then that individual will have Down syndrome. And the same thing happens with the other 2, chromosome number 18 and 13. The final thing under this section is that the age of the parent will make a difference when it comes to the rate at which nondisjunction takes place. As you know from the video lectures on fertilization, female eggs are frozen in prophase 1 of meiosis when the female is still within the womb of her mother, and they're kept in suspended animation until puberty, when one egg is released under normal conditions from an ovary each month. So the older the mother gets, the more likely that this process of nondisjunction will occur. And we've recently found that it also happens in males, but normally at a very late stage, in their 50s, 60s, and 70s. By then, most males have had their children. So it is an age-related factor. On this final slide, let's talk about and go deeper into this process of nondisjunction. Nondisjunction. Okay. So normally what happens is that the chromosomes of the cell, they line up at the metaphase plate during metaphase. And then they are pulled to opposite sides by the cell's spindle. And we assume that the process will share the chromosomes evenly between the 2 daughter cells that result from this process. But sometimes, we'll do that in a different color, chromosomes end up being pulled in one direction only. So that chromosome there, it won't be separated, anaphase, and it'll be pulled towards one pole and not the other. And it depends on when this happens then. So as you know, meiosis is a 2-step process. So we have meiosis 1 and meiosis 2. And understanding the consequence of this type of nondisjunction in each phase is important. So here we can see that the chromosomes are individuals. So they're going to be just sister chromatids. So what will happen in this case is that the two daughter cells produced, let's put it in the same color. So this is one daughter cell, and this is the other daughter cell. This cell will get 4 instead of 5 chromosomes, and this cell would get 6 chromosomes, and that would include 2 red ones, right? So let's put 2 red ones there. Oops. So this one would be red, and there'll be another red one. So this cell on the right is lacking a red chromosome, whereas the one on the left gets both red chromosomes. So from the perspective of this particular species, both cells were supposed to have 5 chromosomes in their gametes, and this one is now n-1, and this one is n+1. The chances are that this will be non-viable, unable to produce an offspring, because missing chromosomes are more dangerous than extra chromosomes. And this one has the potential to produce an offspring with a normal gamete. But as in human cases, they have to be the smaller chromosomes that are accidentally moved to the wrong side. So that's what happens during meiosis 2. So let's look at the situation when it comes to meiosis 1. So let's start again. Let's rehash the drawing, and let's start from the top. So we have a single diploid cell, and let's just work with 2 pairs of chromosomes to keep everything nice and simple. So we'll have a green chromosome pair and a red chromosome pair, okay? And what we're going to do now is we're going to segregate them into gametes, and the gametes should contain 1 green and 1 red. And in this case, something goes wrong during the first stage of meiosis. So as we know, during meiosis, the chromosomes are replicated. So they look like X's. So these are now homologous chromosomes with the sister chromatids. There we go. And then what will happen is they'll line up at the metaphase plate, like just like that. And then one homolog will be pulled that way, and the other one should be pulled the other way. And in this case, the red will be pulled one way, and its partner, the other homologous chromosome, should be pulled the other way. And the result is, the expectant result is this, that we should end up with 2 daughter cells in M1, meiosis 1, and the one on the left will contain a red chromosome, the one on the right will contain a red chromosome, and the one on the left will contain a green chromosome, and the one on the right will contain a green chromosome. So this is what should happen. But if there's non-disjunction at this point, and both, say, red chromosomes get pulled to the same side, let's indicate that with blue, both chromosomes get pulled to the same side, then the situation with respect to our drawing would be such. There will be no red chromosome on the right-hand side, and now there will be 2 red chromosomes on the left-hand side. And then in the next round of cell division, meiosis 2, let's draw that, and that will lead to the production of gametes. You will see what happens. So each mother cell here will then divide further. And in this one here, the chromosomes will be segregated to have 1 green in each one. And on the other side, you'll have 1 green in each one. And now you'll have 2 red ones in each of those gametes. So if this becomes a sperm, then you can see these 2 sperm on the right are n-1, and the ones on the left will be n+1, both those sperm. Okay. So what are the consequences of some of this activity taking place outside of gamete formation? So we do have instances of non-disjunction happening in mitosis. So that's called mitotic nondisjunction. What we just spoke about was meiotic nondisjunction. So in mitotic nondisjunction, it can happen, and the number of chromosomes that are mistakenly pulled to the wrong side can vary from 1 to the entire set of chromosomes. So let's look at the case with the entire set. Let's use a different color, and we're going to now explain mitotic nondisjunction. So here's a normal diploid cell inside your skin somewhere, and it's been asked to undergo cloning, mitosis. So let's assume it's got 46 chromosomes, and those 46 chromosomes will be duplicated and they will line up at the metaphase plate. And in very rare conditions, everything can be pulled to the left and nothing gets pulled to the right. So when this cell divides, the cell on the right has no chromosomes, it's not viable, it'll die. And the cell that's produced on the left will now be carrying double the number of chromosomes. So that would be 4N, or tetraploid. And in some cases, you can have tetraploid tissue naturally. So there are instances, for instance, in the liver, where some of the tissue can do this as a natural means of amplifying the number of cells that can produce products. But this is not normal. So this is an extreme case of all the chromosomes going one way. But normally what happens is most of the chromosomes go the right way, but there's the odd one that doesn't. And that can lead to the production of tissues that contain different numbers of chromosomes. And the term that we use there is that that individual has mosaicism, right? So mosaic tissue. So that can be seen. Let's now turn our attention to the last three terms for this particular slide set. Auto means within, self, and allo means between different individuals. So polyploidy, or a change in the number of ploidy, can happen due to these three mechanisms. The first one's easy to understand. It's very similar to what we just spoke about on the last slide, right? So doing either gamete formation or doing mitotic nondisjunction, you have one cell getting all the chromosomes of the daughters and the other one getting none. So immediately you've doubled the number of chromosomes within those cells. So that would be an example of autopolyploidy. Autopolyploidy. So let's look at an example of alloploidy. In alloploidy, you could have two parents, like two related species of watermelon, in which the chromosomes are still compatible because of evolution. And in one, you could have 4 pairs or 4 sets of chromosomes. So 2n equals 4. So you have a total of 8 chromosomes in that parent. And then a related species, you could have 2n equals 3. That means you have 6 chromosomes in that individual. So during formation of gametes, you'll have the gamete here containing 3 and the gamete here containing 4 different chromosomes. And when you get fertilization, you will end up with a cell that now contains 7 individual chromosomes. And that would be an example of alloploidy. Allo means between two different, sorry, I was going to say self, but allo means between two different individuals. So the two individuals are here, and they will result in the formation of this alloploidy. Okay, and let's now illustrate the last one, the final one for today, and let's clear everything out. So let's talk about allopolyploidy. So allo means between individuals, and let's try to explain this. So one way to get this is you can have, again, related individuals. So you could have 7 pairs of chromosomes in one individual species, and you could have 3 pairs of chromosomes in another. And during reproduction, you will end up with 10 pairs of chromosomes. So that's called allopolyploidy. And in most cases, this individual won't be able to mate with either one of the original individuals. But as we know, with plants and certain other creatures that are hermaphrodites, you can mate with yourself. And in the case of plants, selfing is very common. And that will keep the new species here going, which will be different to either one of those species. Thank you so much, and I hope this very simple chapter contains some wonderful secrets.